So I'm going to try to squeeze 20 pounds of science into a 10-minute sack here. So I'm, let's see how this goes. Okay, so this is the current group. I'm just one of the groups, although I'm responsible for space and infrastructure at the Bulgars campus. I'm just one of the research groups there. I think there are about 10 or 12 independent research groups there now. This is our group. We're active in many areas, and I had a little bit of a. In fact, it's been a long time since I made multiple versions of a talk. So I ended up making, I think, 5 versions of the talk I was going to give this morning, because I couldn't decide what to talk about. And I was going to talk about surgical techniques and devices, but I decided to present very basic stuff. So I hope I don't kill you. Okay. So next slide. Okay, so just as a brief introduction, sorry? Yeah, okay, thanks, Christian. Just as an introduction slide, I hope you guys all know about the multi-scale, multi-level hierarchy of the way that the tendon mid-substance is constructed. It's very interesting, but you have very tightly bound collagen bundles that are and deep inside, you have some. It's a fairly quiet tissue. I was talking with Andy Carr about it this morning. A healthy tendon is very quiet. You have kind of quiescent, meaning kind of sleeping fibroblasts, which are attached to these collagen bundles that are called fibers. These are tightly bound together into the tendon core, the tendon proper. And this is kind of a typical fluorescent stain of the nuclei or the cell nucleus is stained here. You can see that the cells are highly organized along these long longitudinal fibers, and you can see that they're quite intimately attached to the collagen fiber. So we work a lot on tendon disease mechanisms. So I'm interested in tendinopathy, what causes it, how it propagates, and we have. Amongst these people, about half of them are working on hypotheses related to the diseased extracellular matrix, what this means, how it provokes wound healing response, things like inflammation, vascularity or innervation, this neurovascular axis where you have blood vessels where they're not supposed to be. There's a good chance that they're going to be painful because the nerves and the vessels grow in together. And I'm a mechanical engineer who's doing a lot of biology, but so I'm interested mostly in how mechanics are regulating these kind of processes. So I'm going to talk today a little bit just about the mechanics and biology of tendon tears. So this is a nice representation of the different places that tears can happen in the rotator cuff. But basically what happens this is a typical model that we use for an explant, which comes from the flexor tendon of a horse. We can cut out these little pieces of tendon and punch a hole in the middle with a biopsy punch. And we can stretch it, and what you see here is that when we stretch it, you have regions around the tear that are completely unloaded, and the regions that are remaining intact have to carry whatever load these missing regions lose. Okay, so basically you end up with 2 biological regions. You have the unloaded region, which is right adjacent to where the tear is, and then you have an overloaded region, and I just want to quickly say a few things about the unloading. So unloading of the tendon is the model that we use to study that. We use tail tendon from rats or mice. These tendons are nice because they're easy to extract. You can pull them out of the tail without damaging them. And then we can put them into a cell culture condition like this. And basically the tissue in the tail has no vascular supply, has a little one, but it's kind of remote. But you can pull these out, and the tendon doesn't know that it's been extracted from the body actually. And then we can vary the conditions here to kind of investigate how the biology of tendon breakdown works. So when we take the tails out and put them in culture, you can see this is what they look like at the start of the culture, and this is what happens after just even a few days in culture. The collagen structures start to break down, the cells start to round up, the tissues become basically metabolically active. So the tissue starts to turn over. What's interesting is we can vary the culture conditions, and we can model things like a vascularized tendon region or an avascular tendon region. So here we have the full nutrients available to the cells, but we just play around with the temperature and the oxygen. And you can see that the drop in mechanical properties that usually happens, within 2 weeks, typically if you put them in high temperature, high oxygen, or a vascular type environment, the tissue breaks down completely. If you leave them in an avascular condition, the tissue doesn't break down. So we study that, and you can study that fairly deeply. So this is a typical kind of a deep biological analysis of the cell signaling, but you can see that basically these breakdown characteristics are completely different if it's vascular or avascular. So the cells are quiet here, and they become very active here. So that's one side of the equation. The other side of the equation is what happens with overloading. And for the overloading, this is back when I was a baby. I was doing 12 years ago or 14 years ago, I was doing experiments like this where we have a mouse on a little device and we're stretching the Achilles tendon and looking with a fibered microscope at what's going on in the tendon. We can then track the cells as we deform it and start to measure some things on the mechanics side. You can see that these are basically the tissue strains. And although we're stretching the tendon about 30% or 40% even, at the level of the cells are really only being stretched about 2 to 3%. So basically you have these huge deformations in the tissue, the whole tissue, but the deformations at the level of the cells is really small and actually very tightly regulated. So we take it a little bit further, and if we do a partial transection of the Achilles tendon and do the same experiment, what you see is that here is the healthy tendon, the normal intact tendon with the tendon cell strains down around 2%. As soon as we cut the medial gastroc tendon, you can see that the lateral side is then carrying all the load. So we have basically load shunting from the damaged region to the intact region. And this is what you see here. So what you can see is that down here in the functional area of the tendon as a whole, you don't see much change. So although we've removed 30% of the cross-section of the tendon, you don't see much change in the mechanical properties down here on the tendon as a whole. However, on the cellular level, you do. And what happens in this model, if you let it run out, is that the intact part of the tendon will start you'll get appositional growth. It will grow underneath the sheath of the tendon. So this is clearly some sort of anabolic response in the tendon. So we can do kind of similar experiments in the laboratory, and what you see here is we take some tricks from the neuroimaging guys, and we can use dyes where we track the calcium signaling in a tendon. This is a tendon that's not doing much. You can see some background signaling. But what you can see is that when we put this on the microscope and start to stretch it, that basically the cells are feeling what we do, and you can start to see these calcium channels opening up in the membrane of the tendon. So this gives us kind of a window on what the cells are feeling when we do it, and you can see these experiments where we're stretching the tendon over time. This is the stress deformation curves here, and this is the calcium signaling curves here, and the black is where the cells we're counting these calcium events. And what we see is that when we do these experiments, that the threshold for the tissue stretch is around 2% to 3%. So pretty good in line with what we saw in these in vivo experiments on the mouse. Then the question is, what exactly is going on in these tissues? And again, these cells are sitting on these collagen bundles, and the tissues are being stretched. And when we stretch them, the cells are being stretched. They're also being, there's a lot of this kind of motion in the tissue as it's deforming. The cells are also being squeezed, and we can build little devices like this. I'm an engineer, so we build little engineered tendons, and we could put human cells in them, so we could start to move out of the mouse and into humans. We have a biobank here. We take human tendon cells, and we can bring them into these constructs that actually start to look a little bit like tendon. And what we see is that when we stretch these things on the microscope, that we really have to stretch the hell out of them to get the cells to respond. So we have to stretch these things 30% before we start to see the calcium signaling. And this calcium signaling is related to cell death. So when we stretch them that much, the cells start to die. However, when we put them in a similar chamber and put a little fluid flow over them, so we give them a shear stress, we see that the cells are very sensitive to the shear stress. So the stretching, they're not that sensitive, but they're very sensitive to this kind of sliding behavior. And what this might mean, it has maybe some implications, is that when we look at the healthy tendon versus the aged tendon, or a diabetic tendon, there's very subtle but maybe important differences in the way that these tissues behave. So you stretch, the muscle pulls on the tendon, and in a healthy tendon, there's a lot of sliding movement. In an old tendon or a diabetic tendon, the cross-linking becomes very extensive, and the tendons no longer slide. So the collagen bundles no longer slide. So you can see that basically this tissue stretch is taken up mostly as stretch and not as sliding in diabetic tendon and aged tendon. Okay, so Then we could start to, ultimately what we want to get to is a mechanism. So what exactly is going on here? Is this like a drug, something that we can do something with, or is this just nice to know? What's interesting is we can take these model systems that we have and we can start to use pretty advanced biology techniques where we start to knock things out. So what exactly are these sensors that the cells are using to detect these things? And we take these human cells and we knock out a range of possible sensors, and we find that basically there's one sensor that seems to be responsible for this mechanical response. Then there's on the other side, so we know knocking out tends to lose this function, and interestingly there's a mouse model available for a slight gain of function in this. What it is, it's a calcium channel, so it's a stretch-activated calcium channel. You stretch the tendon and these channels open and the calcium comes in. There's a mutation on this gene called piezo1. It's a protein. It's a channel that occurs in the human population, and it makes the channel a little bit leaky. And when we look at mice, the plantaris tendon of mice with this gain of function, you can see that the elastic modulus of these tendons is about 20 to 40% higher. So the diameters of these tendons are exactly the same. So the collagen structure looks the same, the tendon looks the same, but the tendon is about 40% higher, it's about 40% tougher, the failure stresses are higher. It's really maybe important for anabolic response. So, yeah. So what are the implications of this? Basically, it could be a key anabolic trigger. What's interesting is that this gene mutation is under positive selection in sub-Saharan populations. It confers kind of a degree of protection against malarial infection to red blood cells. And it also seems to have effects on the tendon properties. So what we have here is we have a fairly specific target where we suspect, and we have some ongoing human studies to see whether this is having an effect in athletic populations or not. All right, so these are the take-home messages. So tissue deformations are carried by large-scale sliding movements. Cell-level deformations are pretty tight. Shear seems to be a major stimulus for these cells, and we think we've identified at least one sensor for how the tendon cells are detecting it. Tissue vascularity, as you guys know, is playing a huge role in the way tears progress or resolve or don't. Basically, avascular conditions somehow mute the response of the tendon cells, and I think that the inability for this failure to activate the cells is probably behind tear propagation, and that's it. So with that, I'll conclude. Did I take the full 10 minutes at least?
More or less, but it's good.
All right, thanks.
Thanks a lot. Maybe we have time for one urgent question. Yes.
Jess, that was fantastic. Thank you. Really interesting. You've looked at the diabetic group. Have you looked at pathological tendons and perhaps tendons at different stages of the degenerative process?
No, we haven't. But the only thing I can compare that to is that we've looked in cells that we've put into gels where there's basically no tissue structure anymore. And the cells are in a completely different mode. So as soon as they're off of these well-formed collagen structures, pretty much doesn't matter what you put them on, they behave differently.
Yeah.
So I think that if they're quiet and quiescent on the mature tendon structures, they're not doing much. But as soon as we bring them into anything that looks like a wound, they do something else.
One quick subsidiary. We found that there are distinct fibroblast subpopulations, tenocyte. Do you think that's true? So the tendon is not all one type of cell. There are different types of cell, and those cells are doing very different things.
Everything I know about that I learned from you. So.